So we're going to do a crash course of evolution and natural selection. Please remember that this is one of the most tested topics. So while the concepts are pretty straightforward, they are all over the test. Why? Because evolution is the founding fundamental principle of biological systems, and natural selection is one of those major means of evolution. First off, we need to remember that Darwin's ideas of natural selection didn't come out of nowhere. They're based on the work of many previous scientists, okay, looking at things like Hutton and gradualism, Malthus and overpopulation levels. Lamarck is an early evolutionary theorist, but he had some problems we talk about in a bit. Cuvier looked at fossil record, paleontology, sort of catastrophism. Lyell, the principles of geology and uniformitarianism. Okay, while remembering the exact sort of sub-theories is fairly minor, we need to realize that there is sort of this context that Darwin was working with. Within Darwin's actual works, the key ideas are based on some inferences and observations that variation existed, okay, that traits are inherited from their parents, that there are too many offspring to survive, and therefore competition is inevitable. Okay, that's going to tie into many of those previous theorists. But overall, that led to this idea of fitness, or as fitness is differential reproductive success. So the organism that has the most offspring, contributes the most to the gene pool, is the most successful from an evolutionary fitness perspective. In Darwin's seminal work on the origin of species, he defined evolution as descent with modification. Basically, that Earth's many species are descendants of ancestral species that were very different from those alive today. Okay, so that's not our current definition of evolution, but that was Darwin's evolution. He proposed this idea of this relationship that evolution is both a pattern and a process. And in his whole work on the origin of species, the only one that he was actually right about as speciation was his finches, but it's the most common example. Okay, the finches are an example of an adaptive radiation that diversified over time to fill the open niches that they found in the Galapagos. And so there were changes. Those changes were driven by differences in feeding behaviors. Okay, and we see some of that descent over time. But natural selection is one of those forces of evolution. Basically, variation exists. Those variation in traits are inherited, and then that leads to different selective pressures, leading to a change over time. So our current definition of evolution is a change in allele frequency over time. And natural selection is one of the evolutionary pressures that drives that change. Okay, Darwin's finches were a good example of adaptive radiation, but where did that force come from? Well, these birds were coming into the environment. There was no competition, so they began to fill all the niches. And over time, the species filled all of the available niches and created competition within. Okay, that competition led to an overlap in niches. Okay, that overlap led to competitive exclusion, that negative interaction. That then led to character displacement or physical changes in traits due to competitive pressures that then eventually led to speciation. Basically, mutations and differences need to accumulate to such a point that the organisms become reproductively isolated. If reproductive isolation occurs, then speciation can occur. Now, fitness, most traits exist on that polygenic bell curve, but there's three possible results for change over time here. It could be directional, favoring one extreme over the other, like the peppered moth. It could be disruptive, favoring the extremes, like the rock pocket mice. Okay, or it could be stabilizing, favoring the middle, like the number of eggs in the robin nest. Okay, different pressures are going to activate in different environments, which will drive changes. Okay, these can reverse, they can change, they can alter, but selective pressures tend to drive things in particular ways. Natural selection is only one form of selection. Frequency-dependent selection is when the number and the type of phenotype and genotype, okay, that is going to have an impact. Okay, the fitness of any one shape or morph declines if it becomes too common. So you could have a positive frequency-dependent selection where the fitness increases as it becomes more common, or you could have a negative frequency-dependent selection where the fitness decreases as it becomes more common. More commonly, the most common examples here are going to be negative ones, looking for things like genetic variants and infectious disease resistance. That's going to have a negative frequency dependent selection. Okay, sexual selection is going to be driven by mate choice and mate behaviors, non-random mating, and artificial selection is going to be driven by controlled breeding. Basically, humans are involved. Artificial selection is the fastest because nothing is left to chance. Okay, but overall, evolution is a compromise. It's not a goal-oriented process. It's the result of variation, differential reproductive success, and heredity. Just because there's a trend doesn't mean that there's a goal. Traits can be good, traits can be bad, traits can be physical, traits could be social, traits could be psychological or behavioral. Sometimes traits are disadvantageous in certain conditions and advantageous in others. Sometimes, like with sickle cell, there's a heterozygote advantage. Okay, you're resistant to malaria if you're a heterozygote, but if you are homozygous recessive, you have sickle cell, and that's not a good thing. The modern synthesis drives evolutionary change, and we need to realize is that natural selection is just one of these forces. Genetic drift, gene flow, mutation pressure, and all of the various forms of selection all are fundamental to our understanding of what evolution is. Okay, genetic drift is going to be something that's going to be more problematic in small populations. It's random change in frequencies due to chance events. This could be something like the founder effect, or it could be something like the bottleneck effect. Because differences in reproduction exist, traits can change. That could be through some sort of catastrophic event, or through, which is bottleneck, or through the founding of a new population somewhere else, which is founder effect. You can have gene flow and isolation and sort of drive these processes. So if a population shrinks, okay, then that's going to have a major genetic drift. But gene flow is going to allow the exchange and movement of genetic information. So gene flow is going to be when breeding is possible. So if you have a bottleneck, like with the greater prairie chicken, you can bring in organisms from another population, encouraging gene flow to reverse the impact of that bottleneck. Okay. It's all sort of process. The conditions, those traits, are controlled and monitored under the Hardy-Weinberg equilibrium. This is a null hypothesis for evolution. And if you are a diploid organism and you have two traits, then you have P plus Q is equal to 1. The dominant trait, the recessive trait, adds up to 100%. If you then FOIL that out or factor it out, whichever is right, you end up with P squared, 2PQ, Q squared is equal to 1. Homozygous dominant, heterozygous, and homozygous recessive. Okay, those percentages and breakdowns are essential to this process. If they give you the exact count of the heterozygote, count the alleles when you're trying to do Hardy-Weinberg equilibrium. If you have to hypothesize the process, then find Q and move forward from there. The traits, those pressures of evolution, are controlled during Hardy-Weinberg equilibrium. So there is no evolution. It is a null hypothesis. There will be no mutations. There will be random mating, no sexual selection. In fact, there'll be no selection of any type, natural, artificial, whatever. The population needs to be large enough that genetic drift is not a problem, and there needs to be a lack of gene flow. Okay? Gene flow is a stabilizing force, and it will alter evolutionary change. So when you're actually trying to work with Hardy-Weinberg equilibrium, then that's where you're going to get into things like chi-square interpretation. So make sure you take a look at how to do chi-square. Overall, though, evolution is a guiding theory. It's still a theory. People don't really understand what the word theory means, but a theory is a mechanistic process explaining how something occurs. Okay, so it's a big theory. It needs a lot of evidence. Okay, we get evidence from anatomy. Okay, homologous structures are the same because of common ancestry. Analogous structures are the same because of common environments. Vestigial structures are inherited from an ancestor but no longer serve the same function, or in some cases any function, like the pelvis of a cetacean has minimal to no function. Okay, but anatomical evidence can be very useful in determining evolutionary relationship. Sometimes traits are not visible in the adult, and we need to look at the embryo. Okay, something like a post-anal tail or the pharyngeal arches that become the gills in fish. Okay, become the bones of the inner ear in humans. There's different structures that you can't tell in the adult. Sometimes you'll even have traits that throw back to an ancestral trait or an atavism. Okay, so there's all sorts of embryological evidence to look for. Geographical evidence, looking at things like fossil records and distribution of species, that can be very useful. Okay, looking at, say, the sugar gliders and the flying squirrels, this would be an example of analogous traits because they are the same due to similar environment and niche. Okay, you might have traits that are the same because of common ancestry, like with frogs in India and Madagascar because of continental drift, or you might have traits that are shared because of sort of a ring species interaction and biographical distribution leading to isolation and speciation over time. Okay, some traits are going to be controlled by sort of amount of area. So that's sort of looking at the island biogeography effect. The larger the area, the more likely to be diverse. The closer to mainland, the more likely to be diverse. And there's going to be these interactions that we have. Okay, paleontological evidence is going to depend upon half-life, so it's never going to reach zero. You're always going to be losing half every single time. You might look at the bone structure, you might look at fossil record, but the fossil record is incomplete because fossilization is a very relatively rare process. So that's problematic. Okay, biochemical evidence is going to be considered some of our best evidence because the similarities of biochemistry and DNA and amino acids indicate similarities in organisms. So we can see that the human and the horse and the gorilla, okay, the human and the gorilla are very, very similar to each other. Okay, but then I look at the human and the chimpanzee and they're identical. So humans are closest to chimps, pretty close to gorilla, not so close to horses, but horses are pretty close to zebras. That makes sense. You might have traits that are master regulatory genes like homeobox or the Hox genes. Okay, very major differences can happen because of changes over time in the expression of master regulatory genes. We need to keep that in mind. You might have traits that sort of change because of environmental conditions like the resistance to salt in these, I think, oysters. And then you might look at the rate of change and the rate of mutation to develop a molecular clock. So if you have information about how fast mutations are accumulating, you can figure how far back the disease must have emerged. Experimental evidence is increasingly building and then part of the broader structures. So direct experimental evidence like insecticide resistance, that's going to be something that's going to show a great deal of information driving those processes. So if they describe an experiment to you, make sure to read through carefully and break down what's going on. Quick review classification. Remember domain, kingdom, phylum, class, order, family, genus, species. Dumb kids playing catch on freeway, go splat. You are supposed to have from unit 1 an idea of classification, so just a quick review here. Realize that the 3 domains represent eukaryote. That's part of the broader domain that leads to plants and animals and fungus and protists. But then you have archaea and bacteria. Those are going to be the domains of the prokaryotes. The archaeans are slightly closer to the eukaryotes than they are to the bacteria, which we can see here with the archaeans in green and the bacteria in blue. But a phylogenetic tree is going to be a hypothesis of relationship between organisms. Okay, it's going to be based upon differences in genetics. It could be based on changes over time, but a phylogenetic tree is going to be part of that process. You have traits, if you're uncertain, you might have a polytomy, an unresolved difference. You can see that at 0.5, whereas the closest relationships are going to be the sister taxa, which you can see at 0.4. The ones that diverge the most recently are the most closely related. If they give you a scale of axis, then the length of the line matters, like the one on the right. If they don't give you a scale of axis, then it's harder to predict. You might have something that's more like a cladogram, like this one, where the differences are based upon traits. And so everything past the line has that trait. So just be careful and look at what they're marking and what it means. When you're actually trying to compare trees and charts, you have some basic rules. The first is the rule of maximum parsimony. All other things being equal, which is a big assumption, the simplest explanation is true. Okay, characteristics are most likely to evolve only once. So the tree on the left here is the most parsimonious as it involves only one change. The rule of maximum likelihood assumes that mutation rates are equal in different lineages, so more closely related organisms will have fewer differences. So in this case, the tree on the left is evolving at the same rate, so is more likely. Could parsimony be wrong? Yeah. Okay, parsimony is just a way of filtering out these hypotheses, but you still have to test. When you actually get into real organisms, it gets even more complicated, especially when you think about bacteria and archaea. Why? They're capable of horizontal gene transfer, or at least they were long ago, and they still do it now, which means you're going to have exchange of genetic material back and forth, and it's going to get really hard to figure out what's going on at the base of a tree. The chloroplasts and the mitochondria are a great example of that because they were bacteria and they moved into an endosymbiotic relationship with eukarya. So it gets really complicated. When you're trying to figure out speciation or what a species is, we're overall going to use the biological species concept, which says that a species is a species if they are not, if they're basically they're separated by gene flow. If you're able to breed to produce viable and fertile offspring, then you are the same species. Now, that's not the only definition of a species. There are many different definitions. There are actually more than 20. But within this process, we're looking at, okay, well, what prevents that gene flow? Is it because of physical separation? Okay, well, then that's an allopatric speciation. Allo being different, patric being country, physical. Sympatric speciation is going to be some other change that leads to that isolation. What that is, well, we'll look at that in a minute. But there's 22 at least definitions of speciation. That does mean that no one answer is going to answer all instances. So unless they tell you otherwise, we're going to use the biological species concept. But they might pull something else out, so just be careful in what you're reading. Hybridization is going to be the reverse of speciation. So if the separation has not been for long enough, okay, if that barrier to gene flow comes back together again, you're going to create a hybrid zone. And sometimes that species is going to collapse, that hybrid is going to collapse. Sometimes the species will fuse back together into one, and sometimes you'll make a stable third species, but it is possible. Okay, so hybridization is the reverse of speciation, reducing reproductive barriers and varying in the result. Reproductive barriers for sympatric are going to fall into some of these ideas here. You have the pre-zygotic barriers, so that's going to be before sperm and egg come together, and you have the post-zygotic barriers, which is after. Okay, habitat isolation is going to be if they are in different habitats, okay, water versus land, okay, and different bushes, whatever. Okay, temporal isolation, maybe they breed at different times of day or different times of year. Behavioral isolation, maybe they have courtship dances, sort of process sexual selection that they're not successful. If they meet all three of these requirements, if they're breeding in the same place at the same time and the sexual selection has favored them, then there will be a mating attempt. Okay, just because there's a mating attempt does not mean that mating is successful. Okay, so it could be a mechanical problem, basically the parts don't fit, or it could be a gametic problem. The sperm and the egg don't fuse together. That's very common with like ocean-dwelling organisms because they release the sperm and egg broadly. If the sperm and egg are able to come together, then you have fertilization, and now you'll enter the post-zygotic. Remember, you had to produce viable, fertile offspring. So if the hybrid is not viable, if it's really weak and it tends to die before reproduction, that's our first failure. Okay, if they're not fertile, that's the second one, like a horse and a donkey making a mule. Mules do not tend to be fertile. And the weirdest one, the hybrid breakdown, if the hybrid beneficial traits are lost over time, then that's still a reproductive barrier. How is evolution actually occurring? Is it a steady change? Okay, that would be more like Darwin's sort of gradualistic approach, or is it a quick change because of some sort of catastrophic event, like the punctuated model? Okay, short answer is it's probably both. In different times, different events, different pressures can lead to different changes, and it's a bit of a mess. But all of these ideas are coming together into the broader evolutionary pattern that we see. Okay, some of the more common patterns are something like divergent, parallel, and convergent. Looking at these example here, divergent is when they're going to be the same, but they become different. Okay, that would be gray wolf to dog. Okay, convergent, they're the same because of typically analogous features, so dolphin and shark. And then parallel is when two species evolve in response to one another, or coevolution. All of that is possible patterns. Now we've looked at speciation, we've looked at hybridization. The remaining biological pattern is extinction. Okay, extinction is going to be the loss of a branch on that tree of life, where speciation was the emergence of a new branch. You have steady sort of background extinction. It happens all of the time. New species emerge, species go extinct. Most species that have ever evolved on planet Earth have gone extinct. It's sort of a natural process. The background extinction rate sort of is at a level approximate to our normal speciation rate, but because of human intervention, the background extinction rate has gone up, and we are seeing a decline in biodiversity again. If you have sudden large changes in extinction, that's something you would think of like as a mass extinction event. Okay, mass extinction events are these big spikes that we see. Okay, those mass extinction events, massive numbers of organisms die off, and that is something that is incredibly rare. So overall, we say that there have been 5 mass extinction events, and you can see those spikes here. But despite that popularization, there's no definite line separating them from other extinction events. These are just basically the top 5. Mass extinctions can accelerate evolution when the dominance of a particular niche passes from one to another, like with the dominance of mammals after the loss of dinosaurs. But overall, that's going to be that change that we see. Okay. Overall, that's sort of our quick dash into evolution. I hope that made sense and that you are ready for the exam here. If you have any questions, please talk to me.